And now we're bringing the meeting to a close with a real climax, because we started out with Ian Toth talking to us about potatoes and some of the molecular stuff, and now we're finishing on a real high with Jonathan Jones, University of East Anglia, and the Sainsbury Laboratory, giving us a lecture nicely framed around the 3Ds, which is discovery, dissection, and deployment of plant immunity receptors. And I'm sure we'll pick up a lot of the same themes that have been developing during the conference. And I ask you again to populate, as you have been doing, the chat very richly, and we'll use that as a stimulus for the discussion, where we'll take Q&A at the end of the session. So without further ado, over to you, Jonathan, for some of your 12.5% activity.
My 87.5%. Yeah. So thank you very much. Thanks for the invitation. It's a pleasure to be able to talk about our work on understanding plant immune mechanisms and our attempts to deploy that knowledge to a good purpose to reduce crop losses to disease. So you'll have heard this morning about potato diseases. Of course, diseases are not just a problem for potato. And this is from a copy of Science, probably getting on for 10 years ago now, about plant microbe interactions and plant disease problems. Here's a lot of plant diseases that we work on at the Sainsbury Lab, or at least some of which. So wheat stem rust, potato late blight, Asian soybean rust, rice blast, and also wheat blast. And think about it this way. So, if it costs, it's often asserted that on average throughout the world, it takes about 1,000 tons of water to produce a ton of grain. So if you lose 10% of that to disease, that's 100 tons of water you've wasted per ton of grain. So best to bring in sources of resistance. There's other reasons for bringing in sources of resistance, because as is well known, there's a lot of crop protection chemicals applied, and it's been shown that in agricultural fields where there's a long history of fungicide use, populations of mycorrhizal fungi, unsurprisingly, are depleted. So by applying fungicides, we're also impacting crop mineral nutrition. And last but not least, in addition to the obvious reasons, by applying very large quantities of fungicides, we're selecting for fungicide resistance, not just in the plant pathogens, but also in human pathogens like Aspergillus fumigatus, where in the Netherlands there have been cases where spores of Aspergillus fumigatus that have been selected for fungicide resistance have colonized the human lung and basically have been much harder to control with azoles. So if we can protect crops from disease with genetics, with biology rather than chemistry, that's a good thing. And to be able to do so, we need to know what's going on. So this is from a review that I wrote with my colleagues, and indeed, as it turned out subsequently, my co-Wolf awardees for this year, Brian and Jeff. And this sort of attempts to bring together what we've learned in the last 50 years at sort of at a high level. So we know that plants have the capacity to detect apoplastic molecules, pathogen-associated molecular patterns, or microbe-associated molecular patterns, PAMPs or MAMPs, and these are detected via cell surface pattern recognition receptors. And this actually stops disease for most non-pathogenic microorganisms. It keeps them under control. But bona fide pathogens have the capacity to inject into hosts molecules that perturb host biology to make it more conducive for the colonizing organism, and these are called effectors. The defenses activated by these pattern recognition receptors often involve protein kinases. That's the PK that you see in various places here, and sometimes they phosphorylate a transcription factor that affects transcription of defense-related genes. But these effectors interfere with host defense activation. Often they interfere with various components of various protein kinases that are involved in defense. These are protein kinases, for example, that involved in activating a cell surface NADPH oxidase that makes reactive oxygen species. This is potentiated by calcium that activates calcium-dependent protein kinases. But these effectors basically will enable the pathogen to win, unless the effectors are detected by an intracellular receptor. And there's three main flavors. They all have a nucleoside binding site and leucine-rich repeat domain. Some have a coil-coil domain at their end termini, some have what's called a tier domain at their end termini, and some have an RPW8-like domain at their end termini. This is obviously an oversimplification, but these are broad categories. Some of these intracellular immune receptors are sensor NLRs that directly detect pathogen molecules, and some of them can actually oligomerize to make a calcium channel that activates defense. Others don't do this themselves, but they need the action of a helper NLR in order to oligomerize and create a calcium channel. The tier class of sensor NLRs, upon detection of pathogen molecules, oligomerize to a tetramer, which brings the tier domains together, that creates an NADase activity that makes a bunch of small molecules that are detected by EDS1 family proteins, that then, upon binding to these small molecules, associate with a RPW8 NLR, RNL, and Arabidopsis, they're called NRG1 and ADR1. Their oligomerization, upon engagement with activated EDS1 family proteins, also almost certainly makes calcium channels at the plasma membrane, and this is how defense is activated. There's still a big question mark about how these calcium channels actually drive defense, and that's sort of a missing link. But I won't be talking about that, and this is just sort of setting the scene in broad terms. So intracellular NLR immune receptors activate what's called effector-triggered immunity in the jargon. In a way, a better classification would be surface receptor-mediated immunity, intracellular receptor-mediated immunity, SERMI and ERMI, but we don't need to create yet another acronym. So let's stick with PTI and ETI, because those are the ones that are normally used. So I talk about oligomerization. So upon effector detection, individual protomers that have identified an effector change conformation, so they come together and they impose induced proximity on the N-terminal signaling domain. So when these are monomers, basically nothing's happening. But when they come together, four tier domains stuck together, whether it's RPB1, ATR1, 1 ZOPQ, where there's structural information, as you'll see some additional ones, or you'll get this induced proximity of the tier domain. Induced proximity of the CNLs, this one's direct, this one involves an intermediate, there's R1, the first to be found. It imposes induced proximity on the coil domain, and that gives you it's something that can insert in a membrane and create a calcium channel. And this helper NLR from the Solanaceae, NRC4, both the Commun and the Staskowitz labs have shown this forms a hexamer that basically does the same kind of thing, creates a channel. So what am I going to tell you? And because in the tried and tested mode, I'll be telling you what I'm going to tell you, then I'm going to tell you, then I'm going to tell you what I told you. So you have an NLR with, so I'm going to tell you about an NLR that's got broad recognition specificity, and we're trying to understand at a structural level how it detects so many non-identical recognized effectors from a pathogen, and how we've learned that from that to how to engineer it and extend its recognition capacity. I'm going to very briefly tell you that surface receptor mediated immunity, PTI, can potentiate the action of ETI by phosphorylation of a helper NLR. I'm going to tell you something you would expect me to tell you, which is that genetic variation in wild relatives provide a good source of additional immune receptors to enhance disease resistance in crops and the challenges associated with bringing that to public use. And I'm going to tell you a story that's currently in Press and Cell on how co-delivery of helper NLRs with sensor NLRs can extend the range of species in which they work. And also, running through this is we need to be able to use safe and useful methods for crop improvement without excessive regulatory barriers. Right, so here's that slide again. So we're going to tell you about a center NLR of the TRNLR class that is called WRR4. It's an Arabidopsis gene, and it confers white rust resistance genes, actually originally cloned by Eric Holub. And what we found is it recognizes multiple effectors from the pathogen, Albugo candida, an oomycete pathogen, of various and many other brassicacea. Cloned by the fact that Columbia is resistant and to this strain of Albugo, Nidazens is susceptible, standard map-based cloning, and a TRNLR was identified that confers resistance. So what about the recognized entities from the pathogen? So we sequenced Albugo candida, and we defined a variable class of secreted proteins that shared a motif, and the function of that motif isn't known, but it's a CXXC X5G. We call them CCG effectors. And what I'm about to tell you is the work of a talented postdoc, Zhao. But it's based. We started off with data from a postdoc, Amy Redcar, who showed that when we basically cloned a bunch of these CCGs and transiently expressed them with WRR4, this was a search for the recognized effector, and we actually found, to our surprise, there were 8 different recognized effectors for this NLR. So that's where it gets interesting, and I think it's distinct from some of the other studies in the literature. So what are these CCGs? Well, we were stuck for a very long time. Then AlphaFold came along, and we could predict structures for these CCGs, and they actually have multiple ferredoxin-like folds. And ferredoxin-like folds are found in a number of proteins, but they're also found in fungal effectors as well as Albugo effectors. And if we predict the structure of CCG28, 30, 71, 33, and 40 here, they've all got in their N-terminal region a very clear ferredoxin-like fold effector, and that's where the CCG is, post-signal peptide cleavage. And it turns out that you just need this this green bit, for recognizability. So you just deliver 100 amino acids with the WRR4, and you get an HR in tobacco. So from overexpressing and collaborating with structural biologists, particularly in the commune group Selvraj, we now know what this looks like. So this is what the immune receptor actually looks like. Nestling in here is that 100 amino acids or so of the CCG, in this case, I think 40N, 40 N-terminal domain. And there's been a conformational change, brings together these nucleotide binding domains that imposes induced proximity on the TIR domain at the N-terminus, and that's where the enzymatic activity happens. And so this is a collaboration also with Natalia Lukinova in Birkbeck College. So what do we learn from this? So we can see 3 regions where the effector is interacting with the immune receptor. So interface 1 is up here near the LRRs, interface 2 with the middle of the LRRs, and interface 3 involves the so-called CJID domain at the C terminus. And in this region here, you've got amino acids that are polymorphic between the functional allele, Columbia allele, and the non-functional Nidazens allele. And if all we need to do to stop the Columbia allele working is to replace these 2 amino acids with the corresponding amino acids in WRR4, and it doesn't work. That makes perfect sense, because then you'll actually have a charged, two acidic amino acids near each other if you replace the lysine with the glutamate, and you'd have a steric hindrance with this isoleucine compared to a threonine if you mutate that one. So that makes sense to us. But the really interesting thing came when we also did the structural determination for the CCG28 WRR4 complex, and what we found was that actually some of the amino acids in this interface 2 interact with the peptide backbone. So one's used to recognition involving surface exposed amino acids from the receptor and from the recognized molecule, interacting with each other or not. But actually, this interaction is with the peptide backbone. There are a few examples with peptide backbone-based recognition, but not many. They occur when you've got, for example, peptide tRNA hydrolases rely on backbone interactions to enable broad specificity. And if you look at these 3 amino acids here that are interacting with the backbone, this one here interacts with the glycine that is in the C, the G and C, XXC, XXXXXG, and you can see how divergent everywhere else is. So I think we've learned something about how an immune receptor can recognize so many different ligands. And we know that if you mutate this NNQ to alanine, then again, WRR4 doesn't work anymore. So we can predict interactions. It turns out that alpha-fold multimer predicts the structure of the multimer quite well. There's a good correspondence between the predicted structure and the solved structure for where we solved it for CCG40 and CCG28. So then you can look at all of the computationally look for all of the CCGs, and you can see why some of them don't interact. At least they predicted not to interact, and these were negative in that initial screen where we screened about 100. But there are a couple of CCG19 and 45 that actually looked like they ought to interact. So we had another look at them, and guess what? They do interact. So we're now up to 10 different CCGs are recognized by this single immune receptor. And by another approach that I won't have time to go into, we put turbo ID proximity labeling enzyme at the C terminus of WR4 and actually infected with albugo, and we were able to detect an additional 2 effectors from albugo that interacted with WRR4. We did this in an EDS1 mutant, so there was enough pathogen growth. And then when we tested those, they also triggered an HR. So we're up to 12 different CCGs are recognized by the single immune receptor. Yeah, here's the predicted structures for ones that for 67 that we didn't actually determine the structure for. So we predicted then, based on this, well, you could say we determined then that this alpha helix and this beta strand were key to the recognizability. So we did an experiment where we kept these constant and then used RF diffusion to generate something that had the same structure but had a completely different amino acid sequence. And we found that that still activated defense. So we were right in thinking that this yellow region here was all that's needed for recognizability. It's just verified with some co-IPs and some HR assays. But what you really like to be able to do is to evolve the immune receptor to recognize something it couldn't recognize before, in this case CCG47. So Zhao basically tried a lot of variables, and of course you can test these out in silico, and from testing a large number of variables, he found that in silico, if you convert this aspartate to an asparagine, this glutamate to lysine, and this lysine to asparagine or alanine, at least the predicted affinity in alpha-fold multimer went up, and guess what? We actually got an HR when we made this allele of WR4A with these 3 amino acid substitutions. We could confer on it recognition capacity it didn't previously have, and in the course of that, we retained most of the others, but we lost one. We lost CCG33 recognition. So we're just trying to write all this up at the moment. So we've got a model for what's going on. So we do know something about the monomer structure. So actually the embark domain occupies the region that's interface 2. When interface 2 is occupied by the effector, it displaces the embark domain. So it can, and then there's an exchange of, I think, ATP for ADP, and then it oligomerizes, and you've got the induced proximity on the T domain. So again, analysis, I'm going to just do this one more time. Yes, I'm going to stop here. So we actually do have structure. It's not high resolution for the monomer, that the inactive monomer, and this is consistent with that determined structure. Okay, so that's WR4. So another more economically important disease than white rust of Arabidopsis is potato-virus Y in Solanaceae, particularly potato. It's very economically damaging because it's aphid transmitted. When it's in your tubers and you're trying to sell seed potatoes, then basically your batch of seed potatoes doesn't meet requirements, and it turns into animal feed. And so it's a big problem for farmers. So we, with colleagues Jacek Hennig and Marta Gretsch-Barron, identified or cloned the gene for RY that recognizes the PVY coat protein. And in fact, it recognizes the coat protein of many different potaviruses: turnip mosaic virus, plum pox virus, papaya ring spot virus, and others, soybean mosaic virus. So and there's data in this paper actually that shows that it recognizes the fold. So we thought we'd try and understand it. So we ran the same kind of pipeline, and we got a tetramer for this PVY coat protein, RY coat protein oligomer. It's not quite as good resolution as a WR4 data, but we can build a credible model for how the eye recognizes the PVY coat protein. So we again, there's a couple of interfaces that we've defined. PVY itself, once it forms virus particles, the recognized bit is buried. But as soon as this enters a plant cell, it unfolds itself, and so you've got free coat protein that can be immediately recognized, which may underpin the extreme resistance conferred by RY. What we'd really like to be able to do is to understand recognition well enough that we could extend its recognition to other viruses. So cassava brown streak virus is not detected by RY, even though it's got a rather similar coat protein structure, although this is predicted, this is determined. And if you look at the polymorphisms between cassava brown streak virus, it's not recognized, and PVY, plum pox, and turnip mosaic that are recognized, we can see that there are a lot of polymorphisms in that interface. So what we're trying to do now is to evolve RY, so that it can recognize the cassava brown streak coat protein, because that might be quite a useful way to control that disease. It's a work in progress. We've also cloned a gene, RLADG, for potato leaf roll virus in collaboration with Mark Ghislaine and colleagues. It's on chromosome 5. It's a BS4, tomato BS4 homologue, and it interacts with the protease domain of the polarivirus. And we've got quite confident structural predictions, and we'll be seeking to extend its recognition capacity to recognize some important sugar beet viruses that are also polariviruses and cause sugar beet yellows. I'm going to tell you something that is quite important, really, if you're thinking about NLR biology, which is that the NLRs are quite a heterogeneous bunch of things. I got onto this because a Chinese Scholarship Council postdoc, Wang Hao, came to the lab, and he was interested in something he'd cloned, which was actually a wheat gene. So identified by just EMS mutagenesis or azide mutagenesis, I forget which. In wheat, he found an autoactive allele. Basically, he found a mutant line that was kind of debilitated, that showed elevated resistance to powdery mildew, but the collateral damage meant you'd never deploy it. And it turned out to be an autoactive allele of a wheat NLR he called WAI3, wheat autoimmune 3. The wild type allele does nothing, but the autoactive form forms oligomers in blue native in non-denaturing gels, where you can see oligo formation just in gels with an epitope tag on the protein you're interested in. The wild type doesn't form these oligomers, and so working with Hajau, we put it on grids, and with Silver Edge could see in negative staining these entities, and enough structures in cryo-EM to solve the structure, and it forms an optima. So I've told you how TRNLRs form tetramers, and SR35 forms a pentamer, and NRC forms a hexamer, and this guy forms optimers. And it is in what's called the CNL G10 clade. So the CNL G10 clade actually includes some old friends, RPS2 and RPS5 and some 2 from Arabidopsis, along with a number of other interesting genes. And this phylogeny and a more recent one from the Commune lab, from a review that I went into in EMBO Reports in 2023 by Mauricio Contreras. What you can see is these things that are called, we're all familiar with this classification of your CNLs and your TNLs and your RNLs. Well, there's bazillions of different CNLs. Here's the RNLs, NRG1 and ADR1, and here's the TNLs down here. Look at the heterogeneity in the CNLs. There's a whole bunch of different ones. The G10 clade is here, is R1, is MLA10, SR50, SR33, the structure for these guys. Here's the NRC clade over here. Here's RPM1, which is not the NRC clade. RPM1's here, and R3A's down here, another early cloned NLR, and we quite like to look at what structure this one forms. So it turns out that when we overexpress RPS2 under conditions where it's autoactive, but we make a mutant at the N-terminus so it doesn't make calcium channels, we can also see optimers. So you'd argue that wheat is turning out to be a good model system if you want to understand Arabidopsis NLR. So this was reported in 1994 out of both the Ausubel and the Staskovitz labs, and it forms optimers like WAI3. So we think that's a characteristic feature of the CCG10 clade of NLRs. Right, so that's what I wanted to say about structure of immune receptors. Actually, I'm looking at the time and thinking I might have to go very fast here. Now, I've talked about. I guess I should allow myself 5 minutes slack because we took a little while getting started. The PTI initiated by surface receptors and the ETI initiated by intracellular receptors are not completely independent from each other. They work together, and they they mutually potentiate. So it turns out that ETI can act to elevate the abundance of components of PTI, such as the RLCKs and the DPH oxidase. PTI is probably the primary source of resistance. The ETI is receptor limited because most R genes are semi-dominant. Anyway, we call this mutual potentiation, and there's 2 back-to-back papers on this from 3.5 years ago now. Big shout out to my former student who's doing great work now in Ken Shirasu's lab, Bruno. So the TRNLRs that I've talked about at the beginning, like WRR4, these tetramers create an enzymatic activity that can create multiple small molecules, ribo-AMP and ribo-ADP, and also these ADR, ADPR, ATP, and di-ADPR, which respectively activate either the EDS1 SAGA101 NRG1 node or the EDS1 PAD4 ADR1 node of plant immunity. And to cut a very long story short, what we found is that during PTI, this guy gets phosphorylated, and that is part of how ETI and PTI work together. For reasons of time, I won't say more about that, but I'm happy to take questions. So this is relevant because to a strategy we deployed to try and address an important disease for the humid subtropics, which is bacterial wilt caused by Ralstonia solanacearum. It's a root-infecting pathogen, and when it gets into the xylem, it gums up the xylem, so then the plant basically doesn't get water up to the top of the plant, and that's where you get wilt. So we took, this is now a construct we made that carries a lot of immune receptor genes on it. 3 here that I'll come back to for late blight resistance, RY I've told you about, for PVY resistance, and these 3 genes for bacterial wilt resistance. So EFR is a receptor, and these 2 genes, and the surface receptor from Arabidopsis that detects bacterial EFTU, a detection capacity that is absent from the Solanaceae. And these 2 genes are from Klebsiella benthomiana. ROT1 detects a very conserved effector in bacteria, the RIP-B in Ralstonia, SOP-Q from Xanthomonas, HOP-Q in DC3000 in Pseudomonas. And GIM2 recognizes POP-P, what was known as POP-P1 from Ralstonia via ZAR1. So we thought if you put 3 together, we'd have PTI plus ETI for Ralstonia resistance. This is the Royal Society funded collaboration with Mark Gislane and Eric McGenby in Kenya. And so here's the control. We made those constructs and we made transgenics. So here's a line that's not inoculated with Ralstonia, and this is what happens when you inoculate with Ralstonia. And this is actually a duplication of this, so you can ignore it. This one here is one transgenic where it wasn't working, and here are 4 transgenics where we got complete resistance to Ralstonia. And this was very satisfying, and we're hoping to take it further. We would have taken it a lot further were it not for chainsaw Elon Musk closing down USAID. But we'll find a way to get this out there to public use. Of course, many of you have been more expecting to hear more about potato late blight, Phytophthora infestans, the OMI seat, that is the biotic cause of the Irish potato famine in the 1840s. The other, of course, being the British refusing to release grain when the Irish ran out of potatoes. So we've gone to various wild relatives. Solanum venturi was an early one, but we do a lot now with this relative, Solanum americanum, because it's a very tractable model system, and it contains a lot of genetic variation for recognition of effectors from Phytophthora infestans, but also from Phytophthora capsicii, Phytophthora palmivora. I won't have time to go into that. I'm going to stick to infestans mostly, at least. So there's of the lines that we work with, there was a few that in detached leaf assays under high humidity conditions in the lab were susceptible to P. infestans, so we could do genetics by crossing those to resistant guys. But in the field, even those are fully resistant to under conditions where neighboring potatoes go down to late blight. So we've cloned, and I won't go through the detail of it, actually at least 4 different recognition capacities that confer resistance, correlate with segregation for resistance, and we clone their recognized effectors. Now, RPAMR1 and AMR3 work really well in potato. These 2 don't, and we're testing hypotheses as to why that is, but I won't have time to go into that. We reported our analysis of the Americanums in a paper came out at this time last year. And so we've used this to try and solve a problem. The problem is potato late blight, which means that farmers usually have to spray about 15 times a year to control the disease. So here's one of our initial stacks, actually, VNT1, AMR3, and AMR1. And we used CSR stands for chlorosulfuron resistance. So this is an allele of a plant gene, acetylactate synthase, in this case deriving from tomato, with a couple of amino acid changes that mean it's not inhibited by sulfonylurea herbicide, so we can use it as selection of transformation. And these are a couple of genes, or a construct from Simplot, which means you get silencing in the tuber of invertase and polyphenol oxidase, which reduces bruising and reduces the accumulation of reducing sugar upon cold storage. And we made a bunch of transgenics, and we identified lines that are basically indistinguishable from Aristarchus, but they're completely resistant in the field, and they're also resistant in storage. So the RPI gene stack tubers, when inoculated, unlike the Piper control, are completely resistant to tuber blight. And I should say this may be of interest to some of you who are trying to use Golden Gate to make big constructs. What we found was that the constructs from Sylvester, the original ones, work fine for a few genes, but if you want to put in 45 kb, they fall apart or they fall over. They're not stable, basically. So we made a Golden Gate compatible PRK290 derived binary vector that you can look up at Zenodo by just searching for Yules and Egan or Yules and VTech. So, we had these great potatoes. We wanted to be able to deploy them. We posed the question to ACRE, the Advisory Committee on Reef Environment, are these Precision Breeding Act compliant? And so we got a long answer in September 2023. I won't ask you to read it. You can look it up. It's quite interesting reading. Anyway, the short answer is no. And they're not Precision Breeding Act compliant. But they're not Precision Breeding Act compliant because they didn't like the fact that we had a tomato chlorsulfuron resistant gene instead of a potato one, because you can't cross tomato and potato. So even though they're 97% identical, this one was not compliant. And also they ruled that this construct here, it's all potato DNA, but we had basically convergent transcription of two different tuber-specific promoters going through a hairpin of PPO and invertase sequences. They said that could not have arisen through traditional plant breeding, which is probably true. So back to the drawing board yet again. So we made a Solanum tuberosum chlorsulfuron resistant allele. We removed the construct for tuber quality. So, we have to carry on with our acrylamide consumption when we eat poorly stored tubers cooked at high temperature. And we added these two genes for potato viruses resistance, RY for PVY and RLADG for leaf roll virus resistance. And all of these genes, we argue, could have been bred in. There's a bit of detail in the argument we make for why AMR3 and AMR1 could have been bred in. You can actually make somatic hybrids between Solanum americanum or Solanum nigrum, which is basically a hexaploid americanum, and potato, and then backcross and see segregation of resistance genes. So we think we can defend the argument that this could have been bred in, these two. And we hope this will be a product in 5 years, at least in England, but not in Scotland. And so maybe the seed industry will just have to move to England. I throw that out to be slightly cantankerous. So we got partners for this, and one of them is this startup company, BioPotatoes, and you can go to BioPotatoes and read all about the commercial vision and the pipeline that we aspire to. A big thank you to Agnes for this, and also Sasha is an entrepreneur in residence who's trying to help us navigate the challenges to making a product. And so, this is a lot of work. You have, basically, to do this, you need to make hundreds of transformants. You need to PCR screen for ones that carry all the genes you want. You need to measure the copy number, and we do this with AtoDNA. So we only take forward single copy lines that have all the genes. We transiently express the recognized entities in all these plants and only take forward ones where all the genes are working. And then we have to, from a single transgenic line, make cuttings that give us at least 18 per each line. We have to keep track of that. And then these have to be propagated in vitro and then in the glass house, and then they have to be transplanted out. So that was what it looked like in 2024. So what it looks like in 2025. So all these wonderful people planted out in the field about 1,300 potatoes. A big thanks to them. Here they all are. Here's Agnes leading the team, and this is for the International Day of the Potato. So the final story I want to tell you about is a further extension of our capacity to identify useful immune receptors in one plant and move them to another. So back in 1999, the Staskovitz lab published this gene called PEPPER-BS2. So BS2 confers recognition of bacterial spot. It detects AVR-BS2 from Xanthomonas, which will cause this disease in pepper unless you have the BS2 gene, in which case it's controlled. And the AVR-BS2 is a very widely conserved Xanthomonas effector. And it turns out if you take the PEPPER-BS2 gene to tomato, you get the same result, which is good because actually in tomato there's very little variation for control of Xanthomonas disease. But what he did was, what he found that this is the paper from 1999. If you take the BS2 gene and put it into Arabidopsis, it doesn't work. So it only confers resistance. When they tried it in Solanaceae and in Arabidopsis, it didn't work in Arabidopsis, and the lab called it restricted taxonomic functionality, or RTF. So roll on 20 years, and the Commun lab discovers that many sensor NLRs, and this includes BS2, require the NRC family of helper NLRs. So this is from Qihang Wu in PNAS, and this is a really important paper. And they found that various sensor NLRs will work with these three primarily helper NLRs, and they have preferences. So AMR1, for example, and also what plays with NRC2 and NRC3, but it doesn't really function with at least Benth NRC4. AMR3 can play with all of them. BS2 can play with all of them, but they all require at least one of them. And so the obvious hypothesis is, gee, I wonder if you take the sensor NLR and the corresponding helper NLR and move that together, does it break the restricted taxonomic functionality? So one illustration of the success of this idea is work from a collaboration with Camille Vitek and his colleagues at Two Blades in soybeans. So if you take soybean protoplasts and transiently express, let's say, RPI-MR3 and AVR-AMR3 with either a negative control, this random PRR here, or NRC2, you see luciferase eclipse. If you provide the NRC2, that tells you that AVR-AMR3 and RPI-MR3 are activating cell death if you provide NRC2, but not if you don't. And reciprocally for AVR-AMR1. And RPI-AMR1 doesn't do this with AVR-AMR3 and vice versa. So this is press, but there's a collaboration with Hailong Guo, and he's achieved the most spectacular illustration of this. Maheen in my lab has been advancing work on Arabidopsis, which is in the paper. So here's a rice variety that is susceptible to Xanthomonas strain XOC. It carries AVR-BS2, and this is what susceptibility looks like. But if you deliver BS2 plus NRC3 or NRC4 or all three of them, less so with NRC2, it prefers 3 and 4, you get resistance to Xanthomonas in rice, enabled by a pepper immune receptor. And but if you have a strain that's deleted for AVR-BS2, then these gene nothing. So it's not a constitutive activation of defense. It's effective. It's recognition dependent defense activation. So you can actually get sensor NLRs then from Solanaceae to work in other plant species if they're NRC dependent, provided you co-deliver the right NRC or NRCs. And this could be very useful because RPI-AMR3 recognizes AVR-AMR3 homologs, and the same is true AMR1, AMR5, that are found in many Phytophthora species, including ones affecting soybean, cacao, mango, strawberry, papaya, and others. So this might extend that utility. Of course, people will have to get over themselves with respect to using crops that where genes have been moved from one plant species to another, but there's no technical reason why this won't work. So what have I told you? I've told you a bit about an NLR with broad recognition capacity, WRR4, and at this point, structural biology and AlphaFold are changing our understanding of effector recognition. I mentioned briefly how discovery we've made about how PTI can potentiate ETI by phosphorylation of a helper NLR. I've told you about how we can use genetic variation in wild relatives to source useful receptors to increase resistance in crops, and how co-delivery helper NLRs with central NLRs can extend the range of species in which they work. But of course, to do that, we need to be able to use sensible, benign, and useful methods. I discussed that topic at some length in something I published with the Royal Society about 18 months ago. It's on the Royal Society website, Enabling Genetic Technologies for Food Security, deliberate double entendre, telling you about enabling genetic technologies and also how to enable them. I'd like to thank all of my great colleagues, and for the work that I've talked about. Zhao did most of that structural work. This is taking forward our products, the improved potato lines, and Maheen's involved in that, extending restricted taxonomic functionality story. Thanks to funders, collaborators, and thanks for your attention.
Excellent, thanks, Jonathan. A real state of the art. You up?
Oh, yeah, I can see the screen now. That's good.
Good. Okay. Yeah. So thanks, Jonathan. It's a state-of-the-art walk through those molecular interactions, and I'm struck by the complexity of it that you confidently unpick, and also actually looking at those models, some of the beauty of it. They're quite beautiful things that, when you see them in that 3D function, and obviously the striking thing is the move through to the deployment. So we've got some questions, and Katia, you've put quite a few questions in there. Would you like to ask those verbally rather than me translate them? Could you, or do you want me to choose one and read it out?
Well, thank you so much. That's quite an honor. I think I'll go ahead and choose a couple of questions to pitch to Professor Jones. Of course, I was absolutely fascinated by this talk because this has a lot of parallels with my own PhD project. So I would like to know, we talked a lot about an NLR that could be modified through changing one or a few amino acids to make it compatible with a wider range of effectors. What do you foresee is the pros and cons of this system? Because when I was hearing about that, I was thinking perhaps this would make the host plant more vulnerable to having a lot of autoimmune issues, like activating too easily. What are your thoughts on that? What could be some of the drawbacks of having these NLRs with a wider compatibility?
Yeah. Well, I guess let me divide it into two aspects to start with. So I think it's remarkable that WRR4A can recognize so many. It can recognize 12. So whatever trade-offs are required, WRR4A has managed to accomplish them.
Okay, so
we don't really know how. Mm-hmm. We did find, as I said, that when we made some amino acid substitutions to extend its recognition to CCG47, we lost some others. So in principle, you might expect that a simple trade-off, if you're trying to, let's say, edit an endogene to extend its recognition capacity, you might provide a new recognition capacity but lose an old recognition capacity and end up inadvertently enabling a disease that was previously resisted. Mm-hmm. So you're right to say that anytime you make a mutation, you have to worry about autoactivity. So I think you can rule out alleles that have autoactivity in transient assays, or at least reduce the risk that you have inadvertently created such an allele. Of course, there might be mild order activity that doesn't show up in the transient assay, and you won't find that out until you try and deploy it in a field and discover a 10% yield penalty. And yeah, that is certainly a risk. That can't be ruled out, I don't think. Have I adequately addressed those questions of yours?
Thank you for discussing that. I could imagine that if you had this more sensitive and you put it out in a field trial, perhaps some otherwise harmless related species, like some other type of fungus, if it came into contact with a relatively harmless fungus, it might suddenly trigger an autoimmune response. But yes, that often happens in nature anyway. But I think that that's a balanced approach, and I think that screening these plants at all stages of their development is very important to see what the pros and cons.
Yeah.
But I think it's a wonderful idea, and I really enjoyed seeing your data from the alpha fold. Yes, like actually looking at the sites up close. That was fascinating.
I mean, the next 10 years, I think, is going to be about understanding more and more recognitions, atomic resolution. We'll need a lot of new structures determined, but it's remarkable how effective AlphaFold was. But I could add that the AlphaFold was a lot less effective with RY at detecting the structure that we determined. So it's not always going to work.
Okay, thank you.
Okay, thanks. I'm going to come in with a question, and then I'm going to lead into a question Geraint's put up. So I was interested when you were frustrated by the UK blight constructs not being compliant and then rejecting them for what sounded like quite a technical thing. Is it because they've got a set of rules that they just apply, or is it because they had sort of genuine health or environmental concerns about it when it was out there?
It's because they have a set of rules they apply.
Okay.
So the rules are basically the Precision Breeding Act. People have the impression it's all about gene editing, but actually it's about it permits outcomes that could have been achieved by traditional plant breeding. So you could by a couple of point mutations, you can come up with a chlorsulfuron-resistant allele of potato acetylactate synthase, so that's okay. VNT1, RY, RLADG, they're already in the potato gene pool. And the Americanum genes, we are arguing, could also be considered as within the potato gene pool because you can make somatic hybrids and backcross them. Now, so I think they ruled out the previous construct on grounds that they, unless you resort to give a couple of monkeys enough typewriters and long enough they could type the complete works of Shakespeare, you would not have got the construct that was the hairpin for tuber quality by traditional plant breeding.
Okay, so as a follow-up,
I think one could push back on that because there are some events involved in maize domestication that involve inversions and hairpins and the creation of small RNA that has phenotypic consequences on fertility, as Martinson lab has shown. So, but anyway, I just decided that pick your battles. What we could do is come up easily with something that we could get over the line by doing what we've done.
Okay, and so Geraint's question, which I was piggybacking on, really was on that question that they went positive, Acre went positive this time, but you had successes elsewhere in the world, and Geraint says, could you say something about those?
Well, so the genes that we've cloned are being deployed by Simplot in the US. If that's what you mean, I think that's the only jurisdiction where they're known to have been deployed. So you can buy GM potatoes that carry blight resistance and indeed the enhanced tuber quality trait sold by Simplot in the US.
Yeah,
I think
that's what I meant, Jonathan. And just say something about the process of getting those potatoes actually into the market. It's moved
right.
from the UK to a company which isn't based here.
Well, so basically, if you've got a gene that you think could be useful, and you've got enough cash in the bank, then you file a patent. I mean, each filing is probably about $250K. So you've got to be confident you're going to get your money back from licensing. And to be honest, I'm not sure we have yet for the MR genes, at least, maybe for VNT1. But anyway, so your intellectual property office files a patent if they think it's worth it, and they do make a judgment based on if they think they'll get their money back. And then there's a licensing activity by the licensing office. And if Simplot wanted to license RPI VNT1 and RPI MR3, and so now they've brought them, and then it's you send them the sequence or you send them a clone, and then it's up to them. And they have to go through the regulatory process in their jurisdiction, in this case the US. So we're having to do something similar now, and of course it's all very new. The Precision Breeding Act only had its sort of secondary legislation approved by the Lords, I guess, a month ago or so. So we can proceed to do that, but there are still some open questions. So, for example, we're supposed to come up with a Maris Piper that is as much like Maris Piper as possible, but then it's got to meet standards of distinctness, uniformity, and stability to be a new variety. So if it looks exactly the same as Maris Piper, then how is the provenance going to be maintained and administered by varietal inspectors who don't have PCR machines to wander around with, and so there are some details like that. So we have to fulfill distinctness, uniformity, and stability criteria. We have to fulfill value for what is it called, VCU, value for cultivation and use criteria. I think we can do that very easily because it's blight and virus resistant. So and Maris Piper isn't. So obviously it's an improvement on what's already out there. And we'll have to also get it trialed in enough locations, but at a big enough scale that we can make the case that there really is no collateral damage in terms of yield. We've got to pick the right line to do that. So this year, of lines we grew last year, it grew last year in the field in Norwich. We've transferred a subset of those lines that looked best to Phil Howell to be cultivating at NIAB, and compared to Maris Piper, and he has to spray with for late blight control to ensure that our lines, without the advantage of late blight, do not have a yield penalty. So then once we've got a line, and the other problem you got is, this year I got a great potato. Next year I got 10 great potatoes. The year after I got 100 great potatoes, etc. So the amplification stage is really slow. Now at the early stage we are collaborating with Green Seed International, who have a mini tuber propagation unit. But guess what? Their mini tuber propagation unit is in Scotland, where the Precision Breeding Act is not approved. So there's all these little things to battle with to bring it out. But I think we're confident we will succeed, actually, and I think we'll be able to have initial moderate scale plantings. We're hoping for 5 years, maybe 6.